Viruses are an unavoidable part of life, but they themselves are non-living. These simple menaces are not one-size-fits-all and come in a huge variety of shapes, sizes, and functions. In fact, their differences imply that they did not arise from a common ancestor of living things on Earth, and may have even evolved multiple times independently of other viruses. But all viruses do have a few things in common. They are all incredibly small, between 20 and 300 nanometers in diameter. They all have a fixed size and do not grow, but are assembled into a fixed shape. They all have some kind of nucleic acid, either DNA or RNA, as their genetic code. And they all have a protein coat called a capsid. They have no cytoplasm or ability to replicate on their own. Viruses rely on their host for replication. The genetic material of viruses can be either DNA or RNA, but even here there are further possibilities. The DNA or RNA could be single-stranded or double-stranded. There could be large variations in the length of the nucleic acid itself, and the molecules may be circular with no ends or linear with two ends. To be released from their host cell, viruses burst forth in a process called lysis. Some viruses become enveloped in cell membranes during this process, but other viruses do not. Animal cell viruses are commonly enveloped, but bacteria and plant cell viruses typically are not. Viruses have two strategies for replication, and some viruses can switch between these strategies. They are called the lytic and lysogenic cycles. In both strategies, the beginning is the same with attachment and insertion of the nucleic acid. From here, the pathways diverge. In the lytic cycle, the virus hijacks the host cell, replicates itself, then bursts out, killing the host cell. In the lysogenic cycle, the viral DNA becomes integrated into the host DNA and is replicated during cell division. For some viruses, like this lambdaphage, it's possible to switch between cycle pathways and go from lysogenic to lytic, causing the cells to burst. The viruses that are released then enter the replication cycle and can follow either the lytic or lysogenic cycles. Lytic cycles kill the host cells, which could lead to a virus destroying an entire population of bacteria, and in turn, risk destruction for themselves. In humans, our immune systems produce antibodies to help flag and destroy viruses in an attempt to prevent death. Let's look at each step of these cycles in a bit more detail. In both cycles, the first step is viral attachment. Proteins at the tip of the virus bind to the membrane proteins on the cell. Then, the nucleic acid must be inserted into the cell. In the case of this lambdaphage virus, viral DNA is inserted. The DNA is then replicated and transcribed by the enzymes of the host cell. The mRNA is then translated during protein synthesis, forming all of the parts of the viral capsid, and the new viruses are assembled. The cell lyses, and the approximately 100 new viruses are released. During the lysogenic cycle, the viral DNA is integrated into the host cell, first by becoming circular. Then the enzyme integrase inserts the viral DNA into the bacterial DNA. The virus really only exists as a dormant section of DNA called a prophage. The cell goes through normal cell division, making copies of the viral DNA in the process. When the virus is in the lysogenic cycle, it is temperate because it does not kill its host. However, it still has the potential to change to the lytic cycle and cause lysis. Genes in the prophage would need to be activated by stimulus to the cell. In humans, the stimulus could be from severe stress or lowered immune function. But there can be benefit to the host cell. Temperate viruses may include genes from previous host cells, which can increase the genetic diversity of the new host cells when the genes are integrated. The cells will undergo binary fission and pass those genes on. Virus origins are an interesting mystery. There is no gene which all viruses share, and they actually are quite different, which suggests that they have multiple different origins rather than a common ancestor. Viruses are also simpler than any cells, which might imply that they evolved first. The trouble is that viruses rely on their hosts to replicate, which makes them obligate parasites. They can't make more virus without the host cell, so the cell must be present. So how did viruses evolve? If viruses evolve from existing cells, there are two hypotheses about how this may have occurred: the progressive and the regressive hypotheses. In the progressive hypothesis, viruses are built up by components of cells that are modified. Since viruses are essentially bits of DNA or RNA and some protein, it's possible that bacterial sequences of DNA called retrotransposons were modified into bacterial DNA or RNA. When retrotransposons are transcribed and translated, they produce enzymes that can do reverse transcription, allowing DNA to be made from RNA and inserted into chromosomes in random positions. This action is very similar to how HIV works. The original viruses may have also used host cell proteins to create their capsids. In the regressive hypothesis, viruses originate from intraparasitic bacteria that have lost many life functions. There are some parasitic bacteria, like chlamydia, which rely on host cells for some life functions. If you extrapolate that the bacteria could lose even more functions, it might resemble something like a virus. There are even large viruses that have some of their own enzymes and larger genomes, like the Mimi virus, which could support this hypothesis. It's possible that viruses could have arisen by both routes, considering their large diversity. Once these viruses came into existence, they started rapidly evolving. New viral generations can be produced in less than an hour, which is incredibly fast. In each generation, there can be mutations, which are a source of genetic variation. RNA viruses don't perform any code checking, so mutations can occur even more rapidly. The natural selection pressure on viruses is also high because the host cells have mechanisms for detecting and destroying the viruses. All of these factors lead to rapid evolution in viruses. Influenza, which you've likely contracted in your lifetime, is an enveloped virus that uses RNA as its genetic material. This leads to rapid evolution because RNA does not error check. Influenza can also be transmitted between species, like birds and humans, which can increase the number of new strains. On the envelope of influenza are two types of protein antigens, hemagglutinin and neuraminidase. They help bind and release the virus to the host cell. There are 18 varietals of hemagglutinin and 9 varietals of neuraminidase, and they can exist in different combinations, and therefore different strains of the virus. H1N1 was the Spanish flu of 1918, and H5N1 is a bird flu. HIV is rapidly evolving because it's a retrovirus, which also uses RNA. The RNA is not proofread before reverse transcription forms DNA from the RNA code. There are also mutations made by an enzyme that converts cytosine to uracil. These lead to incredibly rapid evolution in HIV. It has the highest mutation rate of any virus. If a person is infected with more than one strain of HIV, the viral genes can combine and form a new strain. HIV can even mutate in the host throughout their lifetime, making it very difficult for the body's immune system to fight the infection. Thanks for watching this episode of Teacher's Pet. Don't forget to like and subscribe and follow me on X @SciencePet.